Yep.
What is your rating? Yeah.
Okay, so the way to figure out how I came up with your total score is to take your number correct on your Scantron and times it by 2, and then add up all the points for your essay. And that was out of 150. So you could figure out your grade by taking that number, whatever your total was, divided by 150, and that will give you your percentage, remember. So 90 and above is an A, 80 and above is a B, 70 and above is a C, for example. If you look at the Scantron, you'll note that the correct answers are actually listed. And so, for example, the correct answer for 1 was D. And the correct answers all show up in a single line, but it is, the 2 columns are actually offset. So if the letter is between, like say, for example, 27 and 28, that is the correct answer for 3. Okay, does everybody understand how to read their Scantron? Okay, so what I would do if I were you is I would go through and look at the questions that you missed and mark them on your test, and I would study those questions in particular for the final exam, right? So make sure that you understand which questions you missed and why. And also, remember that the lab practical is on Friday, and it is going to be fill in the blank, but you're going to have a word list that is in alphabetical order. And so you want to make sure that you study for your lab practical. Okay.
And are you opening up the lab first to study?
Yes, today, yes. And Wednesday if you want to as well. There is labs on Tuesdays and Thursdays.
Oh, so yes, it's
Oh, Thursday as well?
Oh, no, Wednesday as well. Okay, Wednesday, yeah. Sorry. There are labs Tuesdays and Thursdays, yeah. Madison.
So how long is the lab going to be open?
Till 2. Yeah. I'm not going to be around though because I have another class. So I'll actually be in this room.
And how long does lab take?
Okay, so I was hoping to have more people here today because I would like to start the lab practical at 10:30 rather than at 10, just because it takes me hours to set it up, and that just gives me a little bit extra time in the morning. So is that okay with everybody here? It just starts at 10:30? Okay, so I'll send out an email so everybody else can get that information as well. It usually takes an hour. Okay, and then we correct it right after. So you know exactly what your score is right after taking the lab practical. Okay. Okay, so we are starting on a new chapter, and this is chapter 17. And previously, this was actually a chapter that was presented in a different section of human anatomy and physiology. So it was actually presented in biology 233, but we kind of rearranged things for those of you specifically who took the biology 112. So we're moving blood into this chapter. And so the rest of the term is on the cardiovascular system, which includes blood and a little bit of immunity, because we're going to start talking about the white blood cells. And then it talks about the blood vessels. So we'll learn about the major blood vessels. That's something that you're going to need to memorize for the next lab practical. And then we're also going to do the heart. So we're going to do a dissection of the heart. And you're going to learn the anatomy of the heart as well as the physiology of blood flow.
What kind of heart? What type? Human heart or?
Sheep heart. Yeah, sheep heart.
So they freeze
I don't think that they're going to have the cadaver opened up for the heart.
Are they
time.
similar to human?
Yes, they're very similar to human. Yeah. Okay, so when we look at blood, we can talk about the functions of the blood. And one of the functions is to transport oxygen and CO2. So this was actually a question off your midterm. So the transportation of oxygen and carbon dioxide, these are considered the respiratory gases, is accomplished by the blood. The exchange of oxygen and carbon dioxide with the atmosphere, that's the respiratory system. So we're going to talk about specifically the red blood cells and how they have special pigments in them that bind oxygen and carbon dioxide and help to transport those. Okay. The other thing is we can also transport nutrients. So, for example, glucose, right? So blood glucose levels are highly regulated, but they really depend upon what you eat and when you eat it, right? So if you go a long period of time without eating anything and then you have a Snickers bar, your glucose levels are going to go really high initially, and then they're going to be, that's going to be regulated by insulin, and it's going to be taken out, right? But we transport glucose, and glucose is necessary for aerobic respiration where we're producing ATP, okay? So transportation of nutrients, but also other substances, including hormones. So you remember that we talked about the different types of glands that are in your body, and the endocrine glands, which include like your pituitary gland, release hormones like growth hormone, and growth hormone travels throughout your body, and then it binds to different targets. And so growth hormone can bind to the epiphyseal plates of the cells in there, and the cartilage cells would undergo reproduction to allow the bone to lengthen, right? So we talked about the significance of hormones a little bit when we were talking about the different types of glands, okay? Another function of the blood is in thermoregulation. Right? And there's two different ways that this kind of works out, is that you can send blood out to the fingers, for example, when they're cold, right? Or you could take blood away in order to keep the core body temperature up, or you could shut blood out to like the surface of the skin when you're sweating in order to get your body, rid your body of heat, right? So this can kind of warm up extremities, right? Or cool down the body when hot. Your circulatory system also plays an important role in your immune function. So, for example, we have antibodies that are found in our blood plasma. So we have antibodies. And antibodies have a special name. These are called immunoglobulins. And globulin means that it is it's a globular protein, so it's a protein that's highly folded and it's in solution. And immuno just means that it is part of the immune system. So this is different than antibiotics, which are chemicals that we get from primarily bacteria and fungi and stuff like that. Antibodies have a particular shape, and they are Y-shaped molecules. And they are produced by leukocytes, which we'll talk about in detail today. So a leukocyte is a white blood cell. Okay, the other function of the body is the maintenance of pH, helps to regulate pH balance. And the reason for this is that when we look at carbon dioxide that is in the blood plasma, it combines with water. So this is carbon dioxide. It combines with water to produce a weak acid, which is carbonic acid. Okay, so this is carbonic acid. It's a weak acid, so it will dissociate, and so that's why I'm putting these double arrows here, which means that this chemical reaction can go back and forth, and it can dissociate into hydrogen ions plus carboxyl ions. So these were hydrogen, and these are called, oh, sorry, this is not carboxyl, this is bicarbonate. Bicarbonate ions. Okay. So bicarbonate ions help to regulate blood pH because they are a buffer. So these bicarbonate ions can actually also go out into the tissues, and you can excrete them in your urine, but it helps to regulate pH balance in the body. And the reason for this is that if hydrogen ions, which is, remember when you talked about pH, higher hydrogen ions means that it's a lower pH, right? So if you have too many hydrogen ions, it combines with this and it comes and forms back to this. If you have too few of hydrogen ions, then hydrogen ions are released, okay? So if we look at the blood pH, it needs to be regulated between 7.35 and 7.45. Okay, so remember that the pH scale goes from 0 to 14. So would this be basic or acidic? Alkaline or acidic? It's alkaline, right? So it's basic. So it's just slightly basic, right? Because remember that 7 is pure water and that is neutral, right? So this is alkaline, slightly basic. This will be really important when we start to talk about actually respiration and the urinary system, because those two things help to also greatly influence the differences in blood pH. Okay, so let's look at the constituents of blood. So we'll look at whole blood. And we're going to, not this week, but next week, we're going to do what is referred to as a hematocrit analysis. And what you do with a hematocrit analysis is they take whole blood, which contains everything, right out of the body, and they centrifuge it. So when you centrifuge the blood, it means to spin it at a high rate, right? And then it separates based upon the weight. right? So the heavier things go to the bottom, and the lighter things stay up on top. So what goes to the bottom is the red blood cells. So the erythrocytes, which are the red blood cells, are on the bottom. So they are on the bottom of the tube. So their percent of the whole tube is actually the hematocrit value. So the percent of the RBCs equals the hematocrit value. So when we look at the differences between males and females, males, it's about 47% plus or minus 5%. And in females, it's 42% plus or minus 5%. Okay. So what that means is males have a slightly higher red blood cell count than females. They have more red blood cells. And one possible reason for this is levels of testosterone. So testosterone seems to influence the production of red blood cells a little bit. We're going to talk about the main hormone that influences it. So males tend to have a little bit higher hematocrit value than females. So in your book, there's a diagram that shows this process, right? So if we spin out the blood, the erythrocytes, and then we have what is referred to as the Buffy coat. So the Buffy coat, located right here, is white in color. And that contains the other blood cells that are in the whole blood. And those together with the red blood cells are called the formed elements. Okay? So when we look at the Buffy coat, this equals the leukocytes and the platelets. Okay. So the red blood cells and the white blood cells plus platelets equal the formed elements. Okay, so the majority of the blood is actually the plasma. So the plasma, as is shown here, is kind of yellow colored. They say it's kind of straw colored, right? And the reason why it's straw colored is that it has lots of proteins in it, and it tends to be kind of sticky, kind of viscous as well. So we can look at the plasma. Okay, so most of the plasma is water. So like 90% of it is water. But within this water, we have things dissolved in solution, including electrolytes. So electrolytes are charged ions, and these would include sodium, potassium, calcium, and bicarbonate ions. They're called electrolytes because they would transmit, if you had these in water, in pure water, electricity would be able to be transmitted through it because it would be able to pass through because of the charges, difference in charge. Okay, so those are the electrolytes. We also have, like I mentioned before, the hormones and the antibodies, but we also have clotting factors. And the big clotting factor we have in our blood is called fibrinogen. And this is soluble in water. This fibrinogen can be converted, if necessary, to a protein called fibrin. And fibrin is insoluble in water. Okay. So when you injure a blood vessel, instead of bleeding continuously, fibrinogen would be signaled, and we'll talk about that pathway. It's a complicated enzymatic cascade. But eventually, fibrin comes out of solution, and it creates a net, and it prevents excessive blood loss. Okay, so we can talk about serum. So sometimes you hear serum, right? The word serum. And this is plasma with fibrinogen removed. So what this means is that the plasma could clot, right? Fibrinogen could come out of solution into fibrin, but blood serum will not clot. Right? So sometimes you can be given blood serum, and one of the reasons why you might be given that is because it contains antibodies, and it could help you fight off infection, right? But in general, that's the difference between the plasma and the serum. Okay. The other things that are found in this include globular proteins called globulins. So these are globular proteins. So anytime you see the word globule or glob, that means that it is a protein rather than something else, like a nucleic acid, right, or a lipid, for example. Okay. We also have lipoproteins. And these specifically are proteins designed to transport lipids. So these transport cholesterol. Right. So cholesterol is a lipid, and so it is not soluble in water. So in order to transport cholesterol in your body, and you need cholesterol, right? You need it to produce testosterone and estrogen, for example. In order to transport it, you need to bind it to a protein, and that's what those lipoproteins are called. And so when you see, for example, HDL and LDL, right? We'll talk more about this when we start talking about arteriosclerosis and some diseases of the arteries. But HDL is high density lipoprotein, and LDL is low density lipoprotein. So it's important to realize that cholesterol, there's no such thing as good cholesterol and bad cholesterol. The lipoproteins function differently, right? And so high density is better. Is that right? High density? Oh, I always get this back. I think high, I always think bad. But I think high density, you want a higher proportion of HDL than LDL. I think I got it right. Okay. So those are just the differences in the lipoproteins. We also have transferrin. So what do you think this transports? Where have you heard the word ferrin before? Iron, right? So iron, like elemental iron, like ionic iron. So we could think about iron, maybe it could be transported like calcium directly into solution as an ion, but iron cannot be transported directly because it's toxic to us. And so this binds iron. So it binds and transports iron in a way that is non-toxic. Where do you think we store iron primarily in our body? It's in the blood, it's in the spleen, but where is it? Like, if you were gonna eat a meal from a cow and you were like low in iron, what organ would you eat? Nope, liver, right? So iron is stored primarily in the liver. I had a sister that was crazy for liver. I don't know why. Maybe she was needing some extra iron. We don't tend to eat liver because liver sometimes can be where parasites are, but the liver can also have toxins in it, right? Because this also functions to filter the blood of toxins. But healthy liver would be, I guess, okay. Okay. So if we looked at the formed elements, right? The formed elements are the cells. So you'll notice here that just in a smear of blood that there are a lot more erythrocytes than there are white blood cells. So erythrocytes are super abundant, right? And you'll notice something else about them in that they kind of look clear, right? They kind of a dark outline and they have a clear center. But notice that there is no darkly staining nucleus. So the red blood cells lack a nucleus, whereas the white blood cells are larger and they have really large nuclei and kind of weird shaped nuclei. And then the platelets, which we'll talk about, are actually cell fragments. And so they actually are just pieces of cells that come off. Okay. So we're going to talk about erythrocytes first. Okay. So these are the red blood cells. They are produced in the red bone marrow, and specifically produced in hematopoietic tissue. Which is the red bone marrow. They have a shape which is called a biconcave disc. Actually, I think it's with a C. Biconcave disc. Okay. So when we look at their shape has a very large surface area. So the surface area would be greater than if it was round in shape. And so this allows them to pick up respiratory gases quicker. So pick up oxygen more quickly, for example. The other thing about them is that they're enucleated, which means they lack a nucleus. And so what this means is that they are not able to reproduce. So we'll talk about the lifespan of a red blood cell, but they're produced, but they can't reproduce themselves. They have to be produced by stem cells that are in the red bone marrow. And the reason why they lack a nucleus is believed to be, the reason is that therefore they can pack more hemoglobin into the red blood cell. Okay, so they're actually able to pack more hemoglobin.
Do they have other organelles?
Not really. They're just essentially filled with hemoglobin. So the other organelle they do not have is mitochondria. So remember that mitochondria is where cellular respiration takes place. And so the thing about these red blood cells is that they do not use the oxygen that they are carrying. So they don't use oxygen that they carry. Okay, so if we look at a diagram, oops, I have my picture here, there it is. So if we look at the diagram of the red blood cell, right? Interestingly, when you look at frogs and fish and amphibians, they actually have, and I think even reptiles, they actually have a nucleus in their red blood cell. And so this seems to be a mammalian trait. So I think it's only mammals have completely lost the nucleus in the red blood cell. And that's because mammals actually, we need, a lot of oxygen to regulate our metabolism. And so we need to regulate our body temperature, whereas like a frog doesn't need as much hemoglobin because it is not using oxygen to produce heat, right? Okay, so this is the diagram of the red biconcave disc. And the thing about this biconcave disc is it's maintained by the cytoskeleton. And the cytoskeleton inside the red blood cell is very flexible. And so when we think about the capillaries themselves are so small that really only one red blood cell can travel through them at a time. And sometimes they have to squeeze and change shape. And so if you can think of this as being very elastic. So like if you squeezed it like this so that it would get through the capillary, then it's gonna, when it gets popped out at the other end, it's gonna come back to its original shape. And so they kind of are able to move around, and you can bend them, right? They're bendy little cells. Okay, so let's talk about hemoglobin. So hemoglobin is what we call our respiratory pigment. It is composed of globin proteins. So it's made up of globin proteins, and the proteins in the adult hemoglobin, we'll talk about fetal hemoglobin in a minute, is that you have 2 alpha globin plus 2 beta globins. So those are Greek letters. So this is alpha, right? And this is actually 2 beta. We'll put beta. Beta, this is beta, right? So we have 4 total globin proteins in the hemoglobin molecule. And then we have a heme group. And the heme group is what has or contains the iron. So we'll put, this is what makes up the hemoglobin components. There are 4 heme groups. So one hemoglobin molecule can bind to 4 oxygen. So the iron binds the O2, right? So if we have one hemoglobin molecule, right, this can bind 4 oxygen. And when we talk about saturation of the hemoglobin, that's the percentages of the hemoglobin that actually have oxygen bound to them. So if your blood is like 100% saturated, that means all of your hemoglobin has 4 oxygen bound. If your blood is only like 90% saturated, then that means that only 90% of the hemoglobin is bound to oxygen. And so the saturation, right? So we'll put like 100% saturation. That means that all the hemoglobin molecules have oxygen bound. Okay, and they all have 4, right? Okay, so when we look at saturation, we can talk about the main function of hemoglobin is to dissociate from the oxygen when it needs to, or to pick it up. So we can talk about its reversibility. So we have deoxyhemoglobin. This is actually not blue. So when we sometimes think about unoxygenated blood, so that would be deoxy, means hemoglobin that doesn't have oxygen bound to it, we think that it's blue, but really it is actually the reflection through our skin that makes it look blue. And so it's just a different shade of red, right? So it tends to be darker red, right? So it tends to be a darker red, right? And then when it combines with oxygen, this chemical reaction can go both ways, and we call it oxyhemoglobin. Okay, so this would be unoxygenated. This is oxygenated. And this means that it binds reversibly. So if we pick up oxygen in one part of the body, like in the lungs, we need to drop it off in another part of the body. So that's the cool thing about hemoglobin, is it behaves differently in different places of the body, depending upon what the other things are going on, like blood pH and CO2 levels and temperature and acidity. Okay. So this is dark red, and this is bright red. Okay. So the other function of hemoglobin is that it binds carbon dioxide. So about 20% of the CO2 in your body is transported, bound to globin proteins. In the hemoglobin. So the important thing to realize is that carbon dioxide does not compete with the binding sites of oxygen, but rather it binds to the globin protein. So it's not binding to the heme centers. Now this is different than carbon monoxide, right? So when we think about carbon monoxide, which is produced when we incompletely combust, so I'm going to put this in parentheses. This is carbon monoxide. Okay, so like if you have a furnace and it's not getting enough fresh air, or if you're like burning stuff inside your house and there's not enough fresh air to come into the combustion process, then you produce carbon monoxide as a byproduct. And this is a poisonous gas. That binds to the same spot as oxygen. Okay. And it does not bind reversibly, right? So it's like permanently bound, right? And so this actually gives the blood a different color. So this actually makes it kind of pinkish, right? So people that have or die of carbon monoxide poisoning have a pink coloration to the skin. Okay. So that would include people that, you know, run their car in their garage to die, right? That would be carbon monoxide poisoning. Okay. So one interesting thing about hemoglobin is that we have different forms of hemoglobin when we're fetuses as when we are adults. So this is just shows the structure. So you need to know, you need to be able to identify, and the next quiz is going to be next Wednesday. So if I pointed to this, you would say, oh, that's a globulin, right? You don't need to know the difference between the alpha and the beta, right? Those are the globin proteins. This is the heme group, and then in the center of the heme group is the iron. So that center little spot in that diagram is the iron. Okay, so globin chains, heme group, and iron. You need to know that diagram. Okay, so one of the really interesting things about the hemoglobin is that when we're fetuses, we have a different form of it. So fetal hemoglobin, which is sometimes written as HbF, has 2 alpha globins and then 2, oh, I can't remember what this is, alpha versus, I just drew a blank, sorry. 2, not beta, gamma, that's right, 2 gamma globins. This is alpha, right? So 2 gamma globins. Okay. So the thing about this fetal hemoglobin is it binds more quickly to the oxygen. So it tends to pick it up more quickly and it holds onto it longer. Okay, and the reason for this is that the blood that is coming out via the umbilical cord to the placenta is oxygenated, and it actually has to take, and is maybe not completely oxygenated because it's actually gone out right through the body, but the fetus has to remove the oxygen from the maternal circulation. So this specifically helps the fetus obtain oxygen from maternal hemoglobin. Okay, so the interesting thing is at birth, we have to get rid of the fetal hemoglobin. So at birth, hemoglobin and red blood cells are broken down really quickly. So at birth, we need to replace fetal with adult hemoglobin. So when we break down red blood cells, we break down hemoglobin. This is called hemolysis. Right, so the breaking down of hemoglobin. And a byproduct of this is a pigment called bilirubin. And so what do newborn babies typically have a problem with? They sometimes have a problem, and it's really, really common, and it has to do with the color of their skin. Jaundice, right? So newborn babies tend to have jaundice. And this is because their livers cannot process all of the hemoglobin that is being broken down. And so we get bilirubin accumulating in the skin. So it causes a yellow tint to the skin. So if you have a baby and it's warm enough outside, right, they tell you if your baby starts to look yellowish, right, you take them out and you let them sunbathe a little bit, right? And the sun will actually help to break down the bilirubin in the skin, right? If the baby's in the hospital or if it's like really bad weather and there's no sun, then they use what are called Billy lights, right? So Billy lights or sunshine is used to break down the hemoglobin, or excuse me, bilirubin. Bilirubin. Okay, so if we look at the difference between saturation of hemoglobin and oxygen. So if we look at the adults and we look at this graph, so this is oxygen concentration. So at 20%, the adult hemoglobin is going to start to release oxygen from the hemoglobin molecule. So if we look at right here, right, 20%. Up here, at 20%, the fetal hemoglobin, if I draw my line, is way up here, right? And so what this means is that low oxygen levels, adults will give off hemoglobin but the fetus will take it up. Excuse me, give up oxygen, but the fetus will take up the oxygen, that's what I meant to say. So notice how this curve is steeper, right, than the adult one. And what this suggests is that at 20%, the mother is giving off oxygen and the baby is picking it up. And so this is what would be necessary, right? If they were exactly the same, there might be a conflict, right? So that's why fetal hemoglobin is so important for the transfer of oxygen from the mother to the fetus. Okay. So let's talk a little bit about erythropoiesis. So poiesis just means the production of. So hematopoietic tissue, red blood cell, poi, right? So this is just the production of red blood cells. So the production of erythrocytes. So we have stem cells, and I'll show you a picture of them in a minute. And the stem cells go through a series of manifestations. They divide, right? And they produce other cells. And at the very end of this line of production, as the stem cells are maturing and are becoming specialized, they produce what is called a reticulocyte. Okay, so what I'm going to put here is this is specialization. And specifically, we have lots of ribosomes, because ribosomes are where proteins are produced, and these aid in the production of hemoglobin. So I'll show you a picture of a reticulocyte in a minute. The reticulocyte lacks a nucleus at that point. So the nucleus has been ejected, right? So we'll gets rid of nucleus as part of its specialization. So it just ejects the nucleus. But the reticulocyte is important because this is the cell that goes out into the blood and then it specializes into the red blood cell. So the reticulocyte then goes into the blood and it becomes a mature erythrocyte. Okay. So the reticulocytes are important because you can use them medicinally to determine if a person is producing enough red blood cells at high enough rates. So reticulocyte counts are used. Right. This is should be 1 to 2% of all red blood cells. Okay, so this determines rate at which blood cells are produced. So if you have a low reticulocyte count, that means that you're not producing red blood cells enough. So there's something wrong. Your body's not producing them at a high enough rate. And so we're going to talk about anemia, but anemia would be a likely outcome of that situation. Okay. So in your book, they have the process. So you don't need to know all the different names for this undergoing specialization. So this is my stem cell. Right? Notice the stem cells have a nucleus and they actually stay, right? And they would divide. So, I mean, you'd have cells dividing. And so we have stem cells that kind of stay in the red bone marrow. So we don't ever run out of stem cells because they're continually dividing themselves and only a few will then undergo specialization. Okay. So notice here that they have here, hemoglobin is starting to be produced, right? And so that's why they've drawn it from going from like a purple color to a pink color. Here, the nucleus has been rejected. And then here we have the reticulocyte. So this is what a reticulocyte would look like inside the blood. Okay? And then that goes into the blood, and then the reticulocytes then in the blood develop as erythrocytes. Okay, so let's talk about the regulation of red blood cell production. So the stimulus for producing that would cause the production of more red blood cells is what is called hypoxia. So hypoxia means that you have low oxygen carrying capacity in the blood. This could be for a variety of reasons. It could be that you've been wounded and you're hemorrhaging, like maybe you have ulcers in your stomach and you've lost a lot of blood, right? Or it could be that, for example, you don't have enough iron and so you're not producing enough hemoglobin. Okay, so there's many reasons why we could have hypoxia. But this is specifically detected, and this is strange, but it's detected by cells in the kidney. So this is the control center for homeostatic balance of red blood cell protection. So that's the control center. Right? So what those kidney cells do is they produce a hormone called EPO. And EPO is short for erythropoietin. So EPO is a hormone. You might have heard of it because it can be synthetically produced and it can be used by athletes to up the production of red blood cells. Okay? So the EPO then travels to the bone marrow via the circulatory system and stimulates red blood cell, I'll just put RBC, red blood cell production. Okay. So if you have this negative feedback mechanism, then you will maintain the proper number of red blood cells that you need in order to have enough oxygen for your cells in your body. So this shows the kidney response. So the stimulus would be hypoxia. The control center of the kidneys, they stimulate the red bone marrow, and then this enhances and increases the production of the red blood cells. Okay, so we could talk about if you have too much EPO. Right? And sometimes this is because athletes use synthetic EPO as a drug to boost performance, specifically aerobic performances like maybe long distance bicycling, long distance running. Okay. Then what happens is you get what is called polycythemia. Poly means too many, so this is too many red blood cells. Polycythemia. So it's like the opposite of anemia, in some cases of anemia, okay? And so the problem with this is that this increases blood viscosity. Viscosity. So that means like how thick the blood is. And so specifically, if you're in a competition and you get dehydrated, your blood actually becomes thicker, and this can potentially lead to blood clotting and strokes and heart attacks. Yeah. It's mainly considered illegal, I believe. So you can actually stimulate. It's kind of interesting. A few years ago, they had a house that they built. I think it was in Oregon. It might have been by Nike. They built this house that simulated living at a really high altitude. Because if you live at a really high altitude, there's not as much oxygen up there to get. And so what your body does is it produces more EPO. So if I'm living at a high altitude, I will have more red blood cells, right? And so it actually stimulated that. So they could be living in a house that had low oxygen levels, and then they could go out and they could compete. And the same thing kind of goes for being in Denver. And if you're a track person, you might train in Denver for a run in LA, because if you go then fly to LA and then compete, your red blood cells you're going to have more red blood cells than if you train. Right? But that's not considered. I don't think that's considered, but the synthetic EPOs. Sometimes people also do what is called blood doping, where they'll take their own blood and store it, and then right before a competition, they put their own blood back in so they have more blood in their bodies, and that's another way to boost oxygen levels in the body. I think that's also considered really safe. It's probably safer than taking EPO, I would imagine.
How would you be able to tell that? Like if someone would put blood back in, how would you be able to tell?
I don't know. Does anybody know how you can tell that? I don't know if you can tell that. Yeah. Okay.
I mean, so is there any other risks associated with like putting more blood in yourself, I guess?
It would boost your blood pressure, and it could possibly cause blood vessels to rupture. It might also put extra stress on your heart because it has to pump more blood. Okay, so let's talk about anemia, which is kind of the opposite of polycythemia. And anemia, is important to realize, does not necessarily relate to just lack of red blood cells. So this means that the blood does not have enough oxygen carrying capacity. Okay. So we have different types of anemia. So one, for example, would be that you would not have enough red blood cells. This could be due to hemorrhaging. So we could call it hemorrhagic anemia. So you can have blood loss internally as well as externally. It could be something that you're not even maybe aware of, like in the case of ulcers, for example. It could also be that you have hemolytic anemia. So hemolytic anemia means that red blood cells are being destroyed for some reason. And sometimes this happens in the newborn, right? So we talked about their red blood cells being broken down too quickly, but sometimes it is in the fetus, and this is due to maternal antibodies. So that we're going to talk about this when we get to the RH factor. So fetus, and I'm just going to put RH factor in parentheses, because we're going to talk about blood typing, and we're going to do blood typing in lab. And so we're going to talk about what that RH factor is. Okay. The second cause of anemia is that you do not have enough hemoglobin. So you have low hemoglobin content. RH. This is RH. I meant to write an H, yeah. Yeah, RH factor. Yeah. So low hemoglobin content. Okay. So this would be like iron deficiency. Anemia. Females, because they menstruate, tend to have a little bit more need for iron than males. Okay. So sometimes they recommend that you take supplements with iron, but there's lots of things that if you have a well-rounded diet, you should be able to get enough iron from your diet. Things like even spinach are high in iron, so it doesn't have to be meat. And then we have what is called pernicious anemia. And this has to do with a vitamin deficiency, and it has to do with lack of vitamin B12. Now, most of us get enough vitamin B12 in our diets. However, in order to absorb it, you have to have the stomach producing a hormone called intrinsic factor. So to absorb vitamin B12, you must have a stomach that produces intrinsic factor. So I guess pernicious anemia is more common in older people when their stomach's maybe not producing the intrinsic factor, and that people that have autoimmune diseases that attack the stomach lining. So if the autoimmune disease attacks the stomach lining, then the stomach cannot produce intrinsic factor. Therefore, it cannot absorb vitamin B12, and therefore it cannot produce hemoglobin.
What's intrinsic factor? What is that?
It is a protein that is produced by the stomach that aids in the absorption of vitamin B12. So they actually say that the only reason you need a stomach, you know, if we think about our stomach, it does hold, you know, large amounts of food, so we don't have to eat as frequently. But you could remove the stomach entirely and you would be okay, except that you wouldn't have intrinsic factor. So if you have to remove the stomach, you have to give people intrinsic factor, because that would allow the small intestine to absorb vitamin B12. So it's the only necessary reason why we actually have to have a stomach to survive. Okay. So that's pernicious anemia. And then the third one is we can have abnormal hemoglobin. Okay. So what was the example of the disease that you might have talked about? Genetic disease?
Sickle cell anemia.
Sickle cell anemia. And sickle cell anemia is due to a defective beta globin protein. Okay. So that means that the red blood cells are sickled in shape instead of a biconcave disc. So your cells, your red blood cells would look like this, right? And they tend to get caught in small blood vessels like capillaries. And so this would increase the likelihood of somebody having a stroke because of the clogging of the blood vessels, okay? It also makes people that have it really, really weak and tired because they don't have enough oxygen being transported to their blood cells. So in your book, they have a picture of that, sickle cell anemia, okay? So the difference, this would be a normal red blood cell. This is the sickle cell anemia. And remember that we talked about this is actually a common disorder in African-American people with African-American ancestry, because the mutation arose in Africa and it provided an advantage for those individuals that were susceptible to malaria. It actually protects, if you're only half of your hemoglobin is this way, it protects against malarial infection. So there's another group of abnormal proteins that are called thalassemias. If I can write that. Thalassemias. Okay, and this is also abnormal. There's many different abnormal ones, but this is just abnormal globins. And this is in European populations, we have thalassemias, okay, which could lead to anemia. So both of these are genetic diseases. Okay. Okay, I think I'm going to stop here a little bit early today, and we're going to start talking about white blood cells and their characteristics. So in your chapter 17, you might want to take a look. And there's also a mastering quiz for chapter 17, which is going to be due on Sunday. So make sure that you take a look at that, and then we'll talk about that on, I keep thinking it's Monday, but it is Wednesday, isn't it? So we'll talk about that next week. Next quiz is next Wednesday.